going to be isolating an enzyme, glutamate oxaloacetate transaminase, from a natural source, that is picard. So previously we used an egg as a natural source. Here we're going to use picard for this enzyme. Overall, the goals of the experiment are to introduce GOT and its structure, think about what the enzyme is, what its structure is like, and to explore a couple of fractionation steps, that is to fraction all of what's in the heart down into a component, in our case GOT. And we're going to use particularly heat denaturation and filtration, and then also salt precipitation, ammonium sulfate precipitation, and also introduce enzyme assays. All right, so why do we need to isolate proteins at all? Well, number one, we need to study proteins. When we need to do that, we need to look at just one protein. Maybe just a few, but almost always just one in isolation. So we need to focus on that, so we need to get pure enzymes. Oftentimes, this is needed for enzyme metabolism, enzyme mechanism, which are a big part of metabolism. So that's important that we have individual enzymes for this. And then also, oftentimes, we get our proteins from tissues, such as a pig heart, or maybe a source like an egg yolk, or from bacteria. Sometimes we grow proteins in bacteria. And so we need to isolate them and get just the protein by itself to study. In our case, glutamate oxaloacetate transaminase is a 9 kilodalton protein. It's a homodimer, right? And the homodimer means that there are two parts that are exactly the same and come together. So each one of those parts is 45,000 daltons, and then it comes together with a 90,000 dalton protein. We also refer to that as 90 kD, or 90,000 daltons. Again, it's an enzyme, and we find this in kidney and bovine cows, but also this is the structure for the bovine, but also very similar to one that's found in pigs, horses. Right, so this is the structure that we have available for a GOT, and many of them across species are very similar. Right, so why is GOT important, whether it's in pigs, cows, or even humans? Well, number one, it's a big part of processing proteins. So when we have proteins and they start to get broken down, we break them down in the body, and the organisms break them down first into amino acids, and then they need to either recycle those amino acids or get rid of them. Now, key thing about amino acids, they have nitrogens, amino groups attached to them. Too much amino groups in the body can actually be toxic, so we have to handle it very specifically and very delicately and make sure we excrete that. And so that's where GOT is part of. It's part of this processing, this oxidative deamination, where enzymes are getting DNA that's removing the amino groups, moving that NH group from the amino acid, and then once that happens, then it can be processed in the urea cycle. Not involved in this, but after GOT, of course, then the carbon skeleton can be recycled, or it can be made into energy via acetyl-CoA. But really, it's in this step we've highlighted that where GOT comes into play, helping to transfer an amino group from one species to the next, and it can go into the urea cycle and be properly, we can get rid of it there, typically as urea in mammals and other organisms. All right, so again, the urea cycle, just to point out, is again where where the nitrogen here ends up after GOT helps to transfer it and get it here into the urea cycle. That's a big connection, and again, way that we can process nitrogen here. Specifically, this is the reaction that GOT catalyzes. So specifically, it removes an amino acid from aspartate and puts it onto alpha ketoglutarate, and here we end up then with oxaloacetate and glutamate. Now, why this is important is that something like aspartate, it needs to get rid of that nitrogen. There's no direct route into the urea cycle per se by there, but the main point is that glutamate is a great glutamate means they can actually carry nitrogen out of the body, and they're actually sort of essential molecules. So that's what GOT does. It actually catalyzes the transfer of this nitrogen group from here. Again, you notice it's missing here, and it gets formed alpha ketoglutarate to make glutamate, which is a natural amino acid that's part of our body. Alpha ketoglutarate is too, in terms of amino acids. So it's that transfer that occurs. Now, if we try to monitor this transfer directly, it wouldn't be easy, right? It's difficult to sort of observe that with most techniques. And that's why we typically like things that are colored or can be observed in the ultraviolet range, or some range where we can detect it with an instrument. This reaction itself is not so easy to do. All right, so we're going to tell you about an assay how to detect that in just a second. All right, so that's really what GOT does and why it's important in general. And then, again, if we want to study GOT for some reason, for the urea cycle inlets or something else, we would need pure GOT to study. So today, in these next two steps, this part 1, experiment 6, and part 7, we're going to work with a couple of steps to help isolate GOT, and these are called fractionation steps. All right, so fractionation is a term where we can fraction all of what's, say, in the pig heart in a cell into various components. Okay, we're going to fraction them. And how do we do that? Well, we do it by a number of ways. There's many ways you can do it. Today, we're going to, in experiment 7, we're going to talk about 3 in particular. And one of them is heat denaturation. Right, so heat denaturing effects, and we thought about this back when we were talking about lactase and how actually if you heat something too high, it can fall apart and not work and actually precipitate out. But proteins have a variety of ways that they can actually, temperatures at which they're denatured. GOT, we know, is slightly more heat stable, particularly in one form, compared to many other proteins. So we're going to try to use some higher temperatures to denature out some of the proteins which have a lower thermal denaturation point than GOT. And so one step would actually be just heating the solution to 70 degrees or so, 70, 72, and allowing other proteins to precipitate out and keeping behind GOT. Oftentimes, in solution, again, proteins have different structures, they have different functions and different properties, and we can add ammonium sulfate. Right? And so again, this is ammonia, NH4, that we've learned about in Gen Chem, and because it's ammonium sulfate, it has a positive 1 charge. Sulfate has a positive 2, and again, we typically don't draw the charges, but just to highlight the charges, this is extremely soluble. In fact, all ammonium groups are fairly soluble, and so if we have this in solution, we can get a lot of it in the solution, lots of ions. And so if we mix a protein solution with ammonium sulfate, what happens is the ammonium and the sulfate there, and they need to be hydrated by water. Water needs to surround those, right? So we need to have an NH4, and to be dissolved in solution, right? So there's going to be some waters that need to come surround it. And because there are lots of ammonia, lots of sulfates, we need lots of water to come around that to solvate. Well, that happens at the expense of many proteins. The water sort of leaves their hydration shell, goes to solvate these ions, and many of the proteins can then aggregate and stick together and fall out solid. And we can take advantage of that. Proteins having different points once they come out of solution to basically get rid of the proteins we don't want and to capture the protein that we do want, in this case, GOT. And then another one we've seen use some chromatography used before. We're going to use CM chromatography, cation exchange. We're going to hopefully stick our GOT to the column, let other proteins wash out, and then recover our GOT using column chromatography. So these are 3 types of fractionation. There's 3 ways in which we can actually take a solution of lots of stuff and get rid of some things that we don't want, try to retain the thing we do want, which in this case is GOT. And when we are following this, we know, all right, this step did it actually help us with our GOT isolation, whereas GOT not there. And this is where we can use protein assays. Really, assays are tests for the presence
need
of
alucopron after.
So if we have something that's a non-enzyme, it's a protein that maybe does function in the body, an organism somewhere but it's not an enzyme, that's a little bit tougher because it's not real specific. And that's why we need a non-enzyme or a general assay. And that can be something like SDS page. And I just say that we detect any protein, and so we can follow along. Are we getting a protein? Sort of the red one in terms of its weight, and we can follow each fractionation step.
Today?
In graduate school, my protein was just like, didn't have enzyme activity, so we isolated it. We just have to run a gel, we have to look
It's
for it.
closed.
It's not real specific, because something else
It's
could
going to be
because
closed.
weight, but then we might have to do another test to confirm that it's there. That's a non-enzyme. Luckily, GOT
And
is
it's
an
the
enzyme,
opposite direction.
We can observe some change as the enzyme is in the presence of a substrate, and there's a product of it. And we're going to observe some change. I say that because there are a variety of things, whether it's color, UV change, or some other measure.
Okay, bye.
So those are types of protein assays. We're going to use a couple of them. in this particular experiment. One of them is the GOT at a very specific assay. So this top reaction is what we know. This is the reaction we know that GOT catalyzes. Unfortunately, it's not really colored and it's not really easy to notice this in a visible change or anything else. However, if GOT is active in solution and it's catalyzing aspartate and alpha-ketoglutarate to produce oxaloacetate and glutamate, but in this case we're interested in oxaloacetate, what we know is that if we stick oxaloacetate in the presence of malic dehydrogenase and NADH, then we can actually produce malate. Why is that important? Well, NADH absorbs at 340 nanometers, all right, and so that can actually have an absorbance. And then if there's oxaloacetate present from GOT making it and it reacts with NADH via malic dehydrogenase, the NADH will go away. So we'll start off with an absorbance of it, and then as it chews up, that absorbance will go down. And so that gives us sort of something that we can measure spectrophonetically in a spectrophotometer to actually measure that. So we see that. So that's the assay we're going to use, a GOT assay. Also, we know that there's a competing assay, a GDH, or another enzyme, GDH, which will largely do the same reaction, and we're going to test for the presence of it to see if it's in the pig heart that we're isolating. That would actually, if that's presence, then it actually gives us a false indication of GOT activity, and so here we don't want that. All right, so here's the procedure for the experiment. We're going to homogenate the pig heart, and that basically means using a blender, stick in some pig heart tissue with some buffer to help stabilize it and to blend it up. Then we're going to pull out that blended solution. We're going to heat stabilize. We're going to heat denature, sensitive proteins, but we're going to stabilize GOT. And then we're going to test next for what is the best solution of ammonium sulfate, which we just talked about, a developmental assay for GOT to basically isolate it. And then we're going to perform a bulk ammonium sulfate fractionation 1 sample to actually isolate GOT. All right, so that's here for this general procedure for part 1. Again, if we add this heat denaturation step is critical because we don't want to heat denaturate GOT. Right? If we do that, then it's not a solution, it's not working, you can't detect it. So if we add alpha-ketoglutarate and PLP, the enzyme helps to stabilize that. All right, and so PLP is there. We can also add a solution of alpha-ketoglutarate, and we'll do that. We'll add it before we heat stabilize it. PLP is naturally in the solution, so we can take advantage of that as part of it. And so again, it forms this. Notice what the enzyme. This enzyme would be this is GOT. Right? So generally this is the case, but for now our purposes, it's GOT, and it has lysine in the active site and the PLP, which is there, and then we add this enzyme, and notice we get this covalent bond, and we have some other things that are present. And this PLP, along with the alpha-ketoglutarate, which comes in, makes the GOT a little bit more heat stable because of these extra interactions. So that's one of the ways when we get this heat fractionation. Again, adding alpha-ketoglutarate along with the PLP helps to stabilize it. Other proteins are going to precipitate when they don't have that same source of heat, thermal stability, and again allows heat denaturation, as we say, allows them to fall out or precipitate out. We're going to take advantage of that to get rid of unwanted proteins. Then after we do that, we need to make sure that GOT is present, and so we're going to assay for GOT. We just discussed that, and what we'll do is we'll make sure that we have an assay that we're just looking at GOT, and then one that's very similar, using a slightly different buffer to assay just for GDH. Different conditions allow one to react and not the other. When we do this, however, we're going to have to make sure that we dilute. Why? GOT is actually fairly concentrated, at least for this assay, in pig heart, and so we often need to dilute this. And so we'll often talk about a 1 to 160 or a 1 to 80 dilution for these fractions. And so just to let you know, for a 1 to 160 dilution, what we're going to do is if we have a sample, say fraction 1, and it gets diluted 1 to 160, FR1, and we take 1 microliter of that sample, and we would add 159 microliters of buffer. And so it's a 1 to 160 dilution because originally the GOT made up one part of that solution, and so then it's a big part of it, and then it gives 160 of its original concentration. So it's really dilute, and we need this because enzymes can catalyze reactions really quickly, and so we need a small amount of it. And just based on history, we go to the 1 to 160 dilution.
Yes.
So as we begin this procedure, one of the things we did is we actually did have a pig heart, and so from the pig heart we got one of these from a slaughterhouse, and of course this is a smaller one, so we actually dissected. So you can see here what's remaining. So, but the meat of the pig heart has already been cut up, and there's some of it in the beaker right below.
What is the
so we're
next
going to
step
continue
for
with the
the
pig heart to actually
MCAT?
isolate an enzyme from it, the GOT enzyme.
Just sign up for
All right, so this is some of the
So do you just sign up? Yeah, let us start
so we have about 100 milliliters by volume of pig heart here, and we're going to put the rest of it, and here we have another 100 milliliters in there, and we're going to blend this up to basically break apart the cells. And so to do that, we're going to add some potassium malate to help stabilize the pig heart, and it's ice cold, so we're trying to keep all of the components cold, because as they warm up, then that will actually allow them to break down bacteria to grow, those types of things. All right, so we're just going to blend it up for a minute or so. So I think we're blended up, and we'll be back in just a second to show you some of the blended material. All right, so we're back. So we blended up pig heart, and it looks like this, maybe a little bit like a tomato smoothie or something, but so it's all blended up. We measured out exactly 75 milliliters of what we blended up. All right, so what was in here and here, and that's going to be our fraction 1. So FR1 is we're going to call that fraction 1, and that's going to be a total of 75 milliliters. Now we're going to take 5 milliliters of that, and we're going to keep that back, and we're going to do some experimentation with it. But then once we have our FR1, the rest of FR1, fraction 1, we're going to pour into this 250 milliliter centrifuge. This is, again, the total amount is 75 milliliters for fraction 1. Keep a portion of it to do some experiments with, and this one we're actually going to heat up. All right, so we're going to put it into this water bath, which we already have set to 70 degrees Celsius, and we're going to heat it for 20 minutes, and we're going to heat it in the presence of this alpha-ketoglutarate, and alpha-ketoglutarate is going to be able to stabilize. All right, so we're going to just measure that. We want to get out any little remains that we can of the pig heart. This, make sure we do this. We're going to measure out 15 milliliters. We're going to swirl it around. And so then add alpha-ketoglutarate to here. Recall that alpha-ketoglutarate is going to heat stabilize the enzyme by forming the pyridoxal bond with it. And so going to give it a little bit higher stability, and as such, it won't denature. However, we're hoping that many of the enzymes that are in here that are heat sensitive will actually denature, and they'll precipitate out to the bottom of this flask. And if they do so, then that will actually effectively separate them from our glutamate oxaloacetate transaminase enzyme that we're actually aiming for. Right, so that's the thing that's in the pig heart that we want. All right, so we're going to heat this for 20 minutes. It looks like we have it right at about 72 degrees, and we don't want to go above 72 degrees, so that is great. Actually, 75 is the maximum, but 72, and we're going to heat this for 20 minutes. So it's been 20 minutes. We don't want to go more than 20 minutes, and we don't want to go higher than 75 degrees, so we've had it at between 70 and 72 the whole time. And now, since we've heat stabilized the GOT, and we've heated it, we should not denature much of the GOT, but hopefully we denature some of the other ones. However, we want to make sure that we cool the whole thing down so that, one, that we can bring the temperature down for GOT to help maintain it, but also we want to make sure that any proteins that are denatured or maybe slightly denatured, we cool those down and those can precipitate out. So we're going to, again, ice this down for another 20 minutes. All right, so we've iced now for 20 minutes, and so we're back. And so this, again, is our fraction, we're going to call fraction 2. Because remember, fraction 1 was the 5 milliliters that we took out and measured. And so again, fraction 1, it was 5 milliliters plus the entire 70 mils that we put in here. So the total, remember, fraction 1 is 75 milliliters in total. We took 5 milliliters of it to work with. We have now heated it, and so once we've heated it, now we're going to refer to this as fraction 2. All right, so we have fraction 2 again. I have our GOT in here. You notice it's kind of clunky and chunky, and so a lot of proteins and other cell debris that is here. We need to clarify that. That is, we need to get rid of that as much as possible. So we're going to use something fairly standard and fairly routine in terms of cheesecloth. The routine in terms of everyday life,
recording
in the
is
kitchen, we
going
use
right
cheesecloth.
now.
And we're just going to pour this fraction that we've heated to denature a lot of other enzymes, and we're going to pour through here. And it's effectively just going to be a really simple filter. Simple but effective. It's also, and it's kind of low tech, but it is actually highly effective, and we don't need filter paper or anything else like that. And you can just see it dripping straight through. And
So
we're going
I
to
have-
allow that to drip, and we'll have some material left over. So it's going to take a little bit in the top. And we're going to see again, there's still water, and still there's water base,
15.
and so it's going to drip through there. Right? And so we're going to, once it all drips through, we'll measure that, and we'll get a volume, and that will be our true volume for fraction 2. All right, so we're going to call this F, once it drips through, FR2, fraction 2. Recall again that fraction
So
1
I
was
can
really the
just
entire
read it
75 milliliters
instead
that we had
of
earlier. This was a portion of it, and so we actually centrifuged a little bit of it to get rid of any solids, and we took just a little bit of that to make sure it was really pure, and we used the microcentrifuge just to clarify a small amount. You can see we have some reddish solution here, and again, that's part of our FR1. So part of this, we centrifuge this whole tube to get rid of solid, and we centrifuge a little bit more, and then we will use this in an assay, our first GOT assay that we're going to do in just a second. All right, so this again is fraction 1, all 75 milliliters, and split up. We want to think about it as the entire thing that we took out of the pig heart, and then we heat denatured some of that, and we're going to let it drop. It's still dripping a little bit, and so once that finishes, then we'll be able to centrifuge this thing and actually get an assay of this one as well. Okay, so we processed FR1. Remember, fraction 1 was 75 milliliters. We took 5 back and put the other 70 through heat denaturation. After we filtered it, we ended up with 35.2 milliliters of FR2. All right, so that's the fraction 2. We're going to continue that on, but again, still fraction 1. We have part of it, and we're going to work with that a little bit now, where we held those 5 milliliters back, we centrifuge it, and got a little bit of the liquid. Now we're going to test the GOT activity. Recall that the GOT activity is a coupled assay where we're one reaction, and then the product of it reacts by malic dehydrogenase and NADH, and NADH is the source of what we're able to observe, and so we're going to observe NADH here in our spectrophotometer, and our spectrophotometer is set to 340 nanometers, and so that is where NADH absorbs, and then as the NADH reacts, it will degrade, and we'll see that absorbance go down. All right, so an assay really is going to test for an enzyme, and so we're going to test for the presence of GOT. How are we going to do that? Well, we're going to have 2.5 milliliters of our buffer. We're going to put that into a test tube, along with a couple other components. So we have our buffer, and this is the phosphate buffer, pH 7.5, has aspartic acid in it. That's to help select for the GOT. Then we're going to add a couple of other components. We're going to add our secondary enzyme, which is the malic dehydrogenase. There's 100 microliters of that. So mix that in, change tips. All right, then I'm going to add the NADH, and remember that's the source of the spectrophotometer absorbance at 340 nanometers. And then we're going to add our GOT. And again, so this is a dilution of our FR1, and remember we diluted it 101 to 160. Right, so 1 microliter of the fraction 1 supernatant, and we have 159 microliters of buffer, and that's to help dilute it out because GOT is fairly concentrated at this point. All right, so this test tube is about ready to go. However, the one thing that still needs to react is the alpha-ketoglutarate. We're going to add this, put it into the spectrophotometer, and we're going to start a 4-minute timer. And this is, by the way, it's 200 microliters of the alpha-ketoglutarate. And so I find it easy to mix it up with a pipette, and we're going to mix it a couple of times, and then I'll pull out 1 microliter to put into the cuvette. We'll add that to the spectrophotometer and then start our assay. All right, so the beginning absorbance is 0.242 at time 0. So 0.242. You'll notice that it started going down fairly quickly, so right away, but I mean, it's not, it's degrading. Again, we want it to be fairly linear, as best as possible, over about a 4-minute time period. Right, so we're going to observe this for 4 minutes, taking data points every 30 seconds. And again, really, we're most interested in the overall change after 4 minutes, but what we can learn. So it was 0.234 at 30 seconds, 0.234 at 30 seconds. All right, at a minute, it is 0.225. At a minute and a half, it is 0.216. At 2 minutes, it's 0.205. At 2.5 minutes, it is 0.194. At 3 minutes, it's 0.181. And at 3.5 minutes, it is 0.167. And at 4 minutes, it's 0.163. Right, so that was a 4-minute assay of GOT activity. The goal of that was to get a curve that's relatively linear over 4 minutes. That is, basically activity continued over that 4 minutes. And we roughly saw that in this particular case. And so we know, one, that we have GOT activity, or what resembles GOT activity. And so that's a good thing. We also need to check next for GDH activity. Remember that catalyzes the same reaction. But hopefully, if we're lucky, none of it's there, but it has possibility. All right, so for now, though, we've got a GOT activity. So we've done GOT activity on FR1. Next, we want to perform GDH activity on FR1. The reason for this is that the GDH catalyzes a very similar to the same reaction, and so it can impact the activity. So what we want to do is test for GDH activity, and if there's some, we would need to subtract that 1 from the GOT activity, and 2, we would need to continue to monitor the GDH activity. It's really whether GDH is there in this first assay is really dependent on the pig heart. Sometimes it's there, and sometimes it's been activated just in the whole process. So we're going to try for that. Our assay is going to be very similar to last time for the GOT, except we change buffers. We just have a phosphate buffer, and that permits us to have allow the GDH to react. And really, last time we used the aspartic acid, which helped us select for GOT. And we're going to add our MDH. Remember, the MDH is malic dehydrogenase, and that is an enzyme that is there for the coupled part of it. We add our NADH, our source of absorbance at 340 nanometers, and then also we add a little bit of our GOT dilution, 160 dilution parts. Right, and so we're ready to go. Then we need to add 0.2 milliliters of alpha-ketoglutarate to this. We mix it up a little bit, make sure it's even, and then pull out a microliter that we can put into our spectrophotometer. And again, our 4-minute timer. So beginning, the 0.0 is 0.238 is the absorbance. At 30 seconds, it's also 0.238. At 1 minute, we'll call 0.237. At a minute and a half, it's also 0.237. At 2 minutes, it is 0.236. And at 2.5 minutes, it's also 0.236. 3 minutes, it's looking good if we have 0.235. 3.5 minutes is also 0.235. So as we wrap up our 4-minute assay, it looks like the final value is going to be 0.234. So as we've seen those, we noticed that for the GDH, there's a very small change, very small delta. And so this effectively means there is not much, if any, at all GDH. Right? So for the purposes of this, what we're going to say is that change was so small, it would make it insignificant to the terms of the total activity. Where we really get concerned is if that the GDH activity is, some 25% of the GOT activity, or some fairly large number. And this is a fairly small number out in that third decimal of the spectrophotometer. So what we can say confidently is that GDH is not a significant contributor to this activity. And so for the purpose of the rest of this experiment, isolating GOT, we're not going to assay for the GDH. Right? So for scientists, that makes them really happy, right? So they have one less assay to do, but also it just means that there's no contamination. And so far, a couple of things that we can bring in is that our fraction 1, we measured out, we got exactly 75.0 milliliters of it. So it was our fraction 1, so just to give you a sense of the volume. Then we wanted to make sure that we had that 75 milliliters, we wanted to assay the activity of GOT in that, and also get some other metrics, but for now we'll focus on the activity. And so in order to measure the activity using the effect spectrophotometer, again, well, this is the calculation that we do. We look at that change in absorbance at 340, and we talked about that, and over 4 minutes. Again, this is an extinction coefficient, so this is important for correcting for that absorbance and the concentration. Our total solution was 3 milliliters, so we had 2.5 milliliters of buffer. All right, so 0.1 milliliters of malate hydrogenase, 0.1 milliliters of our GOT, 0.1 milliliters of NADH, and 0.2 milliliters of alpha-ketoglutarate to make up a total of 3 milliliters. And then for FR1, it was pretty concentrated, so we need to do a 1 to 160 dilution, as we just talked about. And so again, we were putting in 0.1 milliliters of enzyme, those are GOT. So all of those will help us calculate the activity for these. All right, and so the activity is again in micromoles per minute per mil. So how do we do this? Well, if we remember our GOT at fraction 1, we started off with 0.242. We ended up with 0.163 in terms of our absorbance. All right, that was all over 4 minutes. And then if we calculate and put this in 1.22, and again putting in our units here. And so absorbance really doesn't have units. This is 4 minutes, and then it's a 3 milliliter solution. And then again, 160-fold dilution, and then 0.1 ml. If we do that math, what we end up with is we find we have an activity of 15.2, and then again, this is micromoles per minute per mil. Right, and so that's our activity, and so we know that it is present. We also ran one for GDH, and if you do the same calculation for that, and again, it was a really small change in terms of absorbance, again, because it's present, we can actually calculate then the activity of it. Right? And so based on the difference in activity, I'm fairly confident that we can really just ignore GDH. It doesn't make up a significant portion of the assay activity. Right? But this is how we're able to calculate that. And we did it for FR1 in terms of the activity for GOT and for GDH. One of the other things just to point out at the end of here is just to make sure we show this. So FR2, fraction 2, was the part that we took on and heat denatured. So we had that, and so again, remember that we found out that that was 35.2 milliliters that we had for that particular molecule. All right, so we also needed to develop the ammonium sulfate fractionation. Our goal of this is twofold. Number one, we want to find a level where it precipitates less than 20% of GOT, so getting rid of lots of other proteins, but leaving the majority of GOT. And we're actually going to lose a little bit of GOT, but that's okay because we're going to get back a lot of GOT when we find a percentage that allows us then to recover a vast majority of the GOT. And so both percentages are needed for this second fractionation step. So we had a development of ammonium sulfate fractionation. What we did is create 7 little microcentrifuge tubes. Each one of them we added 0.4 of the fraction 2. And then this first one, we didn't add anything to it. That's really sort of our control standard to see what this looks like. And then we progressively added more ammonium sulfate to increase the percentage of that particular one. Right, and so we got there. And so then what we did is run GOT assays on all of these to figure out which one ultimately is best. All right, so now that we've assayed for fraction 1, we need to continue on to isolate GOT. All right, so our next fractionation step is to try to isolate the GOT using ammonium sulfate fractionation. So really our first step was heat denaturation to try to remove some of the unwanted proteins by stabilizing GOT and then basically fractionating out the unwanted proteins by heat. All right, so that's one. We didn't get rid of them all, we got rid of some of them. So next what we're going to do is ammonium sulfate fractionation. So the ammonium sulfate is a really soluble ion, lots of it in solution, and so because of that, when you put enough of it in the solution, it'll cause some proteins to fall out at various concentrations because the ammonium sulfate is effectively competing for the waters to hydrate them, and they sort of force some water away from proteins, you can think of it that way, and the proteins end up precipitating and falling out. Now depending on the solubility of the protein and its charge and a bunch of other things, this will happen at different percentages for each protein. So we need to find out what that sweet spot is for GOT that we can, one, keep it in solution, right? So add enough of it that a lot of other proteins get precipitated out and it stays in solution. And then another second sweet spot where we add enough of the ammonium sulfate and then the GOT will precipitate out, and then we recover that as a solid. And so if we can do those two things, we can get rid of many other proteins, not all of them, but we still get rid of many other of them, like precipitating out unwanted proteins and then recovering the GOT to continue on. So that's what we're going to do. We're going to have a developmental ammonium sulfate table that you see on the screen. And so effectively what we're going to do in that case is we're going to have our ammonium sulfate. See this? It's saturated, and you can tell that because we have some ammonium sulfate still on the bottom and liquid above it. And so that means we have a lot of that in solution. It's saturated. And so what we've done is each one of 7 tubes, we've added 0.4 milliliters of our fraction 2. We just for fraction 1, test tube 1 here, for this fraction 2, we're just going to have it by itself. That's going to sort of be our standard. And then for each one of the other ones, 2 through 7 tubes, we've added 0.4 also milliliters of the fraction 2, containing some of our GOT. And then we've already aliquoted out the appropriate amount given in the table that we're showing you the developmental ammonium sulfate table about how much ammonium sulfate we're going to add to each one of these tubes, and to basically get to see how much protein is precipitated on each case. So how we do this is we have to add small amounts from here into there to get this to work. So I like to do it in small amounts, about 20 microliters or so, and so I'll pull up some of the ammonium sulfate. Again, that's some of this solution, which we've already aliquoted into these tubes, and then I'll add it to this tube one at a time, and close the cap, and then vortex it. Right? So I'll leave the cap closed, let me know that I've already done that one, and then I will do the same thing for number 3. 20 microliters, vortex it, and continue on. Again, one of the reasons we can't just add it all at once is that it needs to be slow. And also there can be some heat that's with the dilution. So we want to make sure we do this slow so that we can repeat it once we get to a bulk where we're going to take all of our fraction 2 and try to add ammonium sulfate to do the 2 things that I talked about. And so we need to do this slow. So it's going to be tedious, but we're going to take some time to do it. So I'm going to continue on with 4, 5, 6, and 7, adding 20 microliters each time from our tubes where we have that aliquoted off. Each time, picking them up and making sure we stir them. And once I get to the end, I'll start over and keep adding 20 microliters. And it'll take different amounts for each one of these because they have different amounts. But we'll go ahead and continue to do that. And then once we finish that, we're going to incubate them on ice to make sure we cool and allow anything to precipitate. And then we'll centrifuge them in a microcentrifuge to recover the pellet. Right? So I'll show you those results in a second. All right, so as we're finishing up, the microcentrifuge is just spinning down, and so we added the appropriate amount of ammonium sulfate to each one of those 7 tubes, and then we cooled them on ice for 20 minutes, and then we put them in our microcentrifuge to spin. And what you can notice is that if you look at each one of these, there's actually what we call a protein pellet at the bottom, and that is the amount of protein that has precipitated or pelleted out from each sample, given the amount of ammonium sulfate that we put in each one. So we'll pull these out. Each one of them had a different amount, and this allowed us to sort of calibrate the amount of ammonium sulfate that was important in each one. So again, remember the first one should really have no pellet. We didn't add any ammonium sulfate, so that should be our, you notice a little bit of color there. Our fraction 2 and 7 probably should have the most that's here. So what we're going to do with these tubes is that we want to assay the amount of protein that's down here. In particular, we're going to assay or test for the amount of GOT. So in each case, what we're going to do is remove the liquid. We often refer to this liquid as supernatant, so we'll pull it off. We can just discard the supernatant, and we're going to have this solid. What we can do is just redissolve this in buffer, and then we can run our GOT assays on those. All right, so we redissolve this in a little bit of acetate buffer, cellulose, so usually we're going to stick a little buffer in, and then we'll vortex it, get them all redissolved, and then we'll run our GOT assay. Now, this is the same GOT assay that we just showed you a little while ago, using our spectrophotometer, adding the particular ingredients in buffer, and then assaying it over 4 minutes. All right, so we need to do each one of those for 4 minutes. Now, we're not going to show you all of those because it would take a long time, so we're going to show you the data that we're going to have. In fact, you should be able to see that on the screen and to figure out the amount of protein, amount of ammonium sulfate that we're going to add to precipitate out the proteins. So we know an initial sort of cut that we want to add enough ammonium sulfate to our fraction 2 to get a lot of other proteins besides GOT to precipitate out. We can recover that pellet, and then we can move on and add up to 75% ammonium sulfate, and that should pellet out a lot of our GOT. All right, so this is a fractionation step. That is, we're fractionating proteins in various points, and so we're making GOT a little bit more pure at this stage. So based on our calculations, we need 52.8 milliliters of the ammonium sulfate to be added to our 35.2 milliliters of fraction 2. All right, so before we're adding in small amounts, a good thing to do in this case is actually use a burette, where we can actually add ammonium sulfate, 52.8. So when we use up to 50, so we have to add a full amount and add a little bit more. We can open the stop cock, we can actually stir it, and if we do this at a really slow rate, then we can actually add enough of the ammonium sulfate to precipitate out the proteins and get to that 60%. All right, so again, because we need to get to that 60%, since we have 35.2 milliliters of our this fraction 2, then we need to add a little bit more in volume to get to 60%. That's 52.8. So let's go ahead and add that. We'll start adding it, and we'll show you the result. Okay, so we've added the 52.8 milliliters, and then we centrifuged it, and so notice we have the pellet, and a little bit of that other solution still remaining, so we're getting rid of some of the colors, become a little bit more clear. So what we're going to do in this next step is we're actually going to pull off the supernatant. That is, we want the liquid in this case, and now we want to go to 75%. Right, and so when we continue on, we're going to need that 47.8 milliliters to get further to this next step, and that'll get us up to 75%. And so for that, then we can actually have the proteins precipitated out. Okay, so we added all of our volume of SAS, precipitated out the GOT. We actually threw away the supernatant and we redissolved the pellet in 3.6 of the total volume milliliters of sodium acetate buffer. All right, so but in here we have a lot of GOT, but we also have ammonium sulfate, hence the color, and some other proteins. Now, in order for us to work with GOT further, we actually need to get rid of those ions, so sodium and sulfate and any other salts that are in there. That would interfere with our next step, and next step's really to a high salt concentration. So for that, we're going to use dialysis. And so dialysis is a technique where again we have dialysis tubing. This tubing's been manufactured in such a way that has small holes that allows out water, salt, pretty small things. In this case, anything less than 10,000 molecular weight can get out, and then it retains things that are large. So to use this tube is we've been letting it soak, and it's open on both ends, so we need to tie knots in each end, and that'll keep the solution inside. And so tie knot towards the end, make it nice and tight. That allow things not to seep out the end. It's always good to tie a double knot. Sometimes people like clips, but clips seem to leak more frequently, so I think knots are better. So we have one end knotted up, and then we want to put our solution in here. And so I'm just going to use a little funnel, and I like to have that and help pour it in so we don't spill any. And then we're going to just pour our solution, 3.6 milliliters of this, what we call AS2, so ammonium sulfate 2, this portion of it. And so we have that in. And so now we have a lot of extra tubing. That's all right, we just need a small amount for this, but we want to make sure that we have enough space to actually tie 2 more knots. And that's really the thing, is that if we just use a small amount, we wouldn't be able to tie our 2 knots. So I'll tie another one right over there, and those are right on top of one another, so I'm going to tie, just to be sure, 3 or 4 knots is better than 2. And then what we can do is we can just place this in the dialysis solution. And again, this is just more 0.03 sodium acetate buffer, really low concentration, 0.03. And so again, there's some there, it keeps some buffering to allow it to maintain a pH, but also not much salt concentration. So the salt will generally want to come out of the dialysis tubing and into this. And because it's such a large volume, we're talking 3.5 liters, then the salt will be able to dilute out. All right, so this will need about a day. All right, so that'll give us a break at the end of this experiment 6. And then we'll pick up here at the beginning of experiment 7, where we'll take the dialysis solution that has now been dialyzed and more free of the salts, and we'll continue to fractionate GOT. So in this particular experiment, we've been able to start with a K card, chop up some of that K card, add it to a hot plate where it's heated to about 70 degrees, and we thermally stabilized the GOT, and then allowed the other proteins to precipitate out. And then we took that, the solution that we got, fraction 2, and then we added some ammonium sulfate ultimately to it to create a solution where we precipitated out the GOT, and we called that ammonium sulfate 2, AS2. The first one was AS1, we didn't record it. AS2 is what we have here, and we're going to dialyze it, and then next we'll see AS2 dialysis. All right, so if we start to talk about the assays, again, we need to bring in some of the information that we've gathered so far. And so far, a couple of things that we can bring in is that our fraction 1, we measured out, we got exactly 75.0 milliliters of it. So it was our fraction 1, so just to give you a sense of the volume. Then we wanted to make sure that we have that 75 milliliters, we wanted to assay the activity of GOT in that, and also get some other metrics, but for now we'll focus on the activity. And so in order to measure the activity using the effect spectrophotometer, again, this is the calculation we do. We look at that change in absorbance at 340, and we talked about that, and over 4 minutes. Again, this is an extinction coefficient, so this is important for correcting for that absorbance and the concentration. Our total solution was 3 milliliters, so we had 2.5 milliliters of buffer. All right, so 0.1 milliliters of malate dehydrogenase, 0.1 milliliters of our GOT, 0.1 milliliters of NADH, and 0.2 milliliters of alpha-ketoglutarate to make up a total of 3 milliliters. And then for FR1, it was pretty concentrated, so we need to do a 1 to 160 dilution, as we just talked about. And so again, we were putting in 0.1 milliliters of the enzyme, that was our GOT. So all of those will help us calculate the activity for these. All right, and so the activity is again in micromoles per minute per mil. So how do we do this? Well, if we remember our GOT at fraction 1, we start off with 0.242. We ended up with 0.163 in terms of our absorbance. All right, that was all over 4 minutes. And then if we calculate and put this in 1.22, and again putting in our units here. And so absorbance really doesn't have units. This is 4 minutes, and then it's a 3 milliliter solution. and then again 160-fold dilution, and then 0.1 ml. If we do that math, what we end up with is we find we have an activity of 15.2, and then again this is micromoles per minute per mil. Right, and so that's our activity, and so we know that it is present. We also ran one for GDH, and if you do the same calculation for that, and again it was a really small change in terms of absorbance, again because it's present, we can actually calculate then the activity of it. Right? And so based on the difference in activity, I'm fairly confident that we can really just ignore GDH. It doesn't make up a significant portion of the assay activity. Right? But this is how we're able to calculate that. And we did it for FR1 in terms of an activity for GOT and for GDH. One of the other things just to point out at the end up here is just to make sure we show this. So FR2, fraction 2, is the part that we took on and heat denatured. So we had that, and so again, remember that we found out that that was 35.2 milliliters that we had for that particular molecule. All right, so we also then needed to develop the ammonium sulfate fractionation. Our goal of this is twofold. Number one, we want to find a level where it precipitates less than 20% of GOT, so getting rid of lots of other proteins but leaving the majority of GOT. And we're actually going to lose a little bit of GOT. That's okay, because we're going to get back a lot of GOT when we find a percentage that allows us then to recover a vast majority of the GOT. And so both percentages are needed for the second fractionation step. So we had a development of ammonium sulfate fractionation. What we did is create 7 little microcentrifuge tubes. Each one of them we added 0.4 of the fraction 2. And then this first one, we didn't add anything to it. That's really sort of our control standard to see what this looks like. And then we progressively added more ammonium sulfate to increase the percentage of that particular one. Right? And so we got there, and so then what we did is run GOT assays on all of these to figure out which one ultimately is best. Right? So really measuring that. So a couple of things that we did, and there's a lot of data so we didn't show it to you, but to show you what we were doing is that we figured out the percent of GOT recovered. Right? And so then we showed the total GOT in the sample compared to tube 1. And because tube 1 had no SAS attached to it, it should have lots of GOT, and that should really be a high percentage. And then we're focusing this whole assay then what happens in tube 1. So tubes 2 through 7 are compared to tube 1. We can also then measure the protein, number of protein, and compare it. That's one way to do it, but our primary way was doing this. We're actually looking at the activity. You can actually do protein recovered as well. We didn't do that, but we did base it primarily on the activity of GOT because that's what we're specific for. This is a really specific one. It's a little bit more general, so not as much use, but so we use this top one. What we found is when we calculated, and again, we're basing this on the enzyme activity, the GOT activity as we see here compared to tube 1, was that the undiluted, right, so this tube 1 had we're going to call that 100% activity, and then we compared the other tubes to that. Notice that, again, adding all the way up to about 60% kept us below that 20% threshold, which is where we don't get rid of much GOT. Once we get to 65 and 70, then it goes high, and then noticing that once we get to 75%, we're getting well above our 80% threshold. So these are our two values that we like. That is, one of them, 60%, we can add a lot of ammonium sulfate out 60% of the solution and not precipitate out much GOT. Once we get up to 75%, though, most of the GOT is coming out. And so that's actually a good thing, and we see this transition. And again, this graph really helps to illustrate how powerful it is based on GOT activity. So once we got those values, we need to think about how to calculate this. And so for our bulk, because once we know this, then we can add those percentages to the bulk solution of FR2 and actually then be able to isolate then the GOT. Right, so how this works for this, we'll see what it looks like for this 60%. And so we have 35.2 is our volume of this guy. And then again, initially this is 60, or sorry, it's initially 0%, and we want to get up to 60, so this is the difference. So again, volume, so this is where we want to be. This is where we're starting 0 because the protein has nothing, no SAS in it, and we have 1 minus, and then again we want to have 60 divided by 100. And of course, we'll have to forget about dividing by 100 here. And if we do that calculation, we find out that this is 52.8 milliliters is what we would need to add to that 35.2 to get it up to the 60%. All right, and so we'll do that. And then what we found in the end, after that occurred, was that we had a solution remaining once we precipitated out and got rid of the solids of 79. Actually, that's 79.7. Let's make that change. 79.7 milliliters. And that's after we added up to 60%, and now we got to go from 60 to 75%. So that's going to allow us then to precipitate out GOT, most of the GOT, and again, it also doesn't add too much of it, and so that gets us up to about 47.82 milliliters. And this is the SAS that we need to add to the second cut. So again, this is SAS saturated ammonium sulfate. We need to add, and so that's what we added to the second part to precipitate out in. All right, so in this particular experiment, what we've been able to do is observe the pig heart, right, and so observe the tissue, sort of our natural source. We're able to use heat denaturation to isolate some proteins, and we stabilized GOT, and then we used the saturated ammonium sulfate salting out technique to actually be able to isolate GOT. Right? So saturated ammonium sulfate is a salting out technique, and we're able to show how we're able to get rid of most proteins and also ultimately recover GOT from it. A couple of questions as we finish up that I want you to answer in your report. I want you to think about what really is the purpose of the GOT and the GDH assays. How did heating the solution on a hot plate, how did that help with the isolation of GOT? And then third question is, what is fractionation and how was it used in this particular experiment? So this brings us to a close of experiment 6, and we're going to move on to experiment 7.